Hello, as everyone is trickling back, so we're going to start our final session of a very exciting day on biomechanics. And so for this session, we have compiled 4 speakers covering topics such as mechanics of developmental biology to how mechanics regulates tissue homeostasis and mechanobiology, as well as how mechanical considerations can be used to engineer skin tissues. And so our first talk is going to be by Dr. Nadeen. Dr. Nurker is an assistant professor in biomedical engineering, and he will be speaking on mechanics of gut development, I'm assuming.
Okay. I'm not used to this kind of mic. I feel like, welcome to the embryo show. So, in developmental biology, essentially what we're interested in understanding is how you take a single cell and from that create all the complexity and the precise anatomy of a healthy functioning organism. And while this has traditionally been studied as a process of very sophisticated gene regulatory networks that inform cell fate determination, as engineers, we can appreciate that this is also a process of very profound physical transformations as well, right? From the seeming disorganization to the very fully, exquisitely, and stereotyped pattern of our tissues and organs. So why try to understand this? So there's a couple of reasons. The first is we need to understand how this process normally occurs if we want to understand why it doesn't always occur normally, right? What the basis is for birth defects. Can we understand that? Can we screen for that? Can we treat that and prevent it? In addition, the idea of starting with a pool of stem cells and then building organs is essentially the very purpose of tissue engineering as a field, right? And so the idea here is that if we can understand how evolution has solved this problem, then it can serve as a roadmap for tissue engineering and regenerative medicine strategies as well. So that is developmental biology. Now let's talk about mechanics. So in the simplest sense, we can think of mechanics as forces that act on a material that has certain properties, whether it's stiffnesses or viscosities, and that produces a particular movement, a deformation, or a flow. And as seemingly simple as this might be, when we apply mechanics to biological systems, it's not that simple anymore because the forces, the properties, and the movement all involve cells that respond to their surroundings, which means that the forces depend on the stiffness depends on the forces, and so it creates all of these nonlinearities. And what my lab is generally interested in is studying the mechanobiology of development. So how it is that these processes are harnessed and manipulated to create organs during embryonic development. To do that, we rely on the chick embryo, which is a classical model of developmental biology. It's a classical model for several reasons. First, because evolutionarily, birds are very close to mammals, which means the governing principles of embryonic development in chickens are conserved primarily with mammals and with humans. So we can get insights into human development from studying the chick embryo. But the differences from mammals are also very important. So for example, birds, of course, develop outside of the mother, which means you have access to the embryo in a way that's very difficult in mammals. And that allows you to do things like directly observe the deformations during tissue formation. Right? So this is just an example where we can see the cells, we can see the really dramatic tissue deformations that are occurring during the formation. These are the first stages of forming the lungs, the heart, and the upper digestive system. That accessibility also means that you can manipulate forces within the embryo. And so this is just work from a couple of students in the lab where they've developed a method to apply tension on the order of nanonewtons to micronewtons to not only look at the effects of tension on morphogenesis, but also to probe the mechanical properties of tissues in vivo within the developing embryo. In addition to that, the chick embryo is quite large when compared to the mouse and other developmental model systems. And what that means is that we can directly measure physical properties. So we can do, for example, uniaxial tensile testing. This is what you would do if you just had a large slab of some engineered material that you wanted to characterize. And we can do that with living tissues. So those are the tools that are available to us. And now I'll talk about what questions we're aiming to address with those tools. The first is to understand how genetic and molecular cues regulate tissue scale forces to shape the embryo. Right? So how, through all the complexity of embryonic development, do you ensure that the forces are the right forces at the right time, in the right place, with the right orientation, and the right magnitude, acting on tissues of the right stiffness to get the same stereotyped outcome time and time again. And then how do you get robustness in that through feedback? How do the forces actually regulate the overlying biology as well? And then on a longer time scale, if we can understand the biological controls on morphology, can we understand how you manipulate those to create different outcomes, which is exactly what has happened through evolution. So we address these questions in a number of different contexts, and what I'll talk to you about today is how we're addressing these in relation to the organogenesis of the small intestine, which is shown here from a 16-day-old chick embryo. So your intestine is very long, and we might think of it as just this mess of tubing, but in fact it's actually very precisely organized. And it has to be precisely organized to avoid wrapping around itself, as occurs in a number of birth defects. And this organization results in looping of the tube, back and forth, back and forth, back and forth. And what's really remarkable about this pattern is it is conserved for a given species. And so what that means is that if you go into any individual in this room and count how many times their intestine loops back and forth, it is almost identical to everyone else that's here. So that tells you that the mechanics of taking a straight tube and forming this looped organization has to be something that is tightly regulated, right? So a chick embryo will hatch after 21 days of incubation. At 5 days, the tissue that will form the intestine is this approximately straight tube. And by 16 days, it has formed this looped organization. You can see what I was saying in these intestines from 3 different individuals, right? The pattern is almost identical across these 3. You can cut the blood vessel that runs down the middle out, and the structure will linearize, and that allows you to appreciate this very regular periodicity in the organization of the intestine. This is true not only in the chick, but in the mouse, as well as the human, which is shown here from an MRI of a Carnegie stage 21 embryo. So the way you go from a straight tube to this looped organization it turns out to be buckling, a mechanical instability. So this is what happens when you have something that's long and narrow, and you press on it, right? It might shorten a little bit, but primarily what it's going to do is it's going to bow out to the side. The compression that drives that buckling comes from differential growth between the intestinal tube that you can see here and this web-like tissue attached to the tube, which is known as the dorsal mesentery. So initially they have the same length and they're stuck to one another, but over time the tube elongates much faster than the mesentery, and so the mesentery compresses the tube, and that compression is what drives buckling. One way that you can see this is if you just dissect and separate these tissues after buckling is complete, and what you find is that the mesentery retracts and the tube straightens out, right? So we can characterize the morphology of buckling using a wavelength and radius of curvature as a readout of the phenotype, quantified. And the nice thing is that we can predict the wavelength and curvature by measuring just 5 things, all of which are accessible in the lab. The thickness of the tube in the mesentery, the stiffness of the tube in the mesentery, and the relative growth rates of one to the other. And so as a system to understand the mechanics of morphogenesis, this is then very useful because, first of all, the morphogenesis relies on a very simple physical mechanism because it's just buckling driven by compression. All of the mechanics that describe this are directly measured in the lab. And then we can layer that on top of all of the things that have made chick a classical model of embryology, including the ability to misexpress genes and test their gene function. And so you can see, for example, here in previously published work from our group, that we implicated BMP signaling in this process as a core regulator of the differential elongation rates. So that when you have too much BMP, you get too much buckling, which creates midgut volvulus, a birth defect that occurs in humans. And when you inhibit that, you get too little looping, you have a disorganized structure that fails to internalize and gives rise to gastroschisis, which is another one of these birth defects. So buckling is not only involved in packaging of the intestine, it's increasingly recognized as a core physical mechanism of morphogenesis, implicated in placement of the villi within the luminal surface of the intestine, branching of the airways, and even cortical folds on the surface of the brain. And so our expectation, our hope is that by studying the small intestine, where we actually have access to measure all of these things, that we can get insights into the morphogenesis of these other organs and organ systems as well. And so now going back to our mechanics description, we can say our movement is the wavelength and curvature of buckling. We have one molecular handle on the forces, and then we can ask, well, what determines the connection between the two, the physical properties of the mesentery? Because this is really what translates the differential growth into the compressive forces that buckle the tube. And to talk a little bit more about stiffness, it's not a constant for this tissue. So if you look at this plot here, the y-axis is basically how hard you pull on the tissue. The x-axis is how much it stretches. And what you see is that the mesentery has this behavior where it's very easy to stretch it out at first, but then the tissue locks up and becomes very difficult to deform further. So this is a behavior known as constitutive nonlinearity. And if you think about this in the context of buckling, that property is very important because what if that transition from soft to stiff happens too early. Then you have too much resistance to elongation, which leads to buckling too early and too many loops. Whereas if you shift this transition to the right, then you may never generate enough compression to actually drive the mechanical instability in the first place. And so what we're interested in is understanding what actually sets that in an embryonic tissue. We're surprised to find the involvement of cells is actually quite important. So here is a similar stress versus strain plot. So how hard you pull, how much it stretches. For controls, when you treat with a drug that inhibits the ability of cells to contract, the curves shift and collapse down to the left, which means that they stiffen at a much lower strain or a much lower stretch. And this was perplexing to us because we figured if the cells matter at all, then removing their contribution should effectively soften this tissue. But instead it does the opposite, it effectively stiffens it. And so the way that we're thinking about this is described in the analogy here. So you could imagine if you have a rope, which would be, in this case, extracellular matrix fibers like collagen and elastin and things. And if there's slack in that rope, it's very easy to pull the ends of that apart until there's no more slack, and then it becomes very difficult to pull it further. So you could think of slack being extinguished in the soft part of the curve, and then once you're actually pulling on and engaging the fibers, you're dealing with the stiff part of the curve. So add on to this analogy the cells. And our proposal is that cells through contraction are basically creating compressive pre-stress on the matrix fibers, and in doing so are basically providing the sort of mechanical clutch that determines when the fibers become engaged and setting the transition point. If this is true, you'd expect to be able to actually see this when you look at the fibers, and indeed we do. So this is second harmonic generation imaging. This is just a way of being able to visualize collagen fibers in a tissue without fixation, without staining. And in the control, we have these wavy fibers, but when you treat with the drug to remove contractility, those fibers actually straighten out. So then the remaining question that we're trying to answer now is, does this change in structural properties actually, is this actually enough to explain the macroscopic change in mechanics? And I won't go into the details of how we're testing that, but we're using constitutive modeling, which is sort of a classic approach of biomechanics, to ask whether changes in the structural properties can explain the macroscopic changes that we observe in the tissue mechanics. Okay, so going back to our motivating questions, we've talked a little bit about how molecular cues set up the forces that sculpt the embryo, both in terms of differential growth and physical properties of the tissue. I don't have time today to talk about our investigations into mechanical feedback, but we do see that this core mechanoresponsive pathway, the hippo pathway, when it's dysregulated, we can actually force this sort of phenotype where you get a loss of looping. And then I'll just end very briefly with talking about evolution because I think it's fun. So the idea here is, if we can understand the molecular control of forces, the biological control of morphology, as engineers, what we'd want to know is how you can manipulate that to create different outcomes. And that's exactly what evolution has done to create morphological diversity across species. And so the postdoc working on this just got fertilized eggs of like every bird that we could get our hands on. And we just looked for things that were particularly rich phenotypes for getting insights into the mechanics of morphogenesis. I'm just going to show you one of them, but there are several that we were pretty excited about. And that phenotype comes from the intestine of the Long Island or Pekin duck. So most of the intestine looks like the chick, and the human and the mouse and everyone else. It's just undulating back and forth, back and forth. But oddly, a central loop breaks that symmetry and elongates instead of buckling. So it doesn't participate in the normal buckling morphogenesis of the anterior or posterior part of the intestine. And that just becomes more accentuated with time. And so what we can do with that then is then collect this tissue, send it for RNA sequencing, and just look for differences in gene expression that correlate with the presence or absence of buckling. And so what we see is that before this central loop becomes morphologically distinct, that it's quite similar to the anterior and posterior loops that do participate in buckling. But as it grows out and becomes more distinct and fails to buckle, we start to get separation at the transcriptome level. And so what we can then do is look at what it is that's responsible for creating that separation to generate candidates that are novel regulators of tissue mechanics driving buckling in the embryo. And with that, hopefully you just have a nice snapshot of the type of stuff that we do. I'll acknowledge John Durrell, a PhD student in the lab who did all the contractility work, and Elise Lefay, a postdoc in the lab who did all the evo-devo stuff. Thank you.
Great, thank you so much. So we'll be taking questions at the end. So our next speaker is Dr. Hassan Apache. Dr. Apache is an assistant professor in dermatology and BME, and he'll be telling us about engineering skin.
Okay. Hi, everyone. My name is Erbil Abachi. I'm an assistant professor in the Department of Dermatology, and recently I got affiliated with biomedical engineering. And my lab is primarily focused on skin tissue engineering for drug testing purposes and for transplantation. And it's a challenge to engineer human skin given its cellular and structural complexities. In fact, human skin has more than 50 different cell types and many appendages such as hair follicles and sweat glands. It is mainly composed of 3 layers. The bottom layer is the adipose tissue, and then you have the connective tissue, another connective tissue as your dermis, and then finally you have your epidermis, which has also many layers and is housing epidermal stem cells and differentiated layers of the epidermis. And the skin is our largest organ that connects us to the outside world, so it's no surprise that it is equipped with protective and interactive functions. In fact, human skin has more than 20 billion T cells, which is twice the number of cells that are circulating in our blood. It is highly innervated with different specialized sensory neurons that specifically sense itch, heat, pain, cold, or touch. So given this complexity, it's really fascinating for tissue engineering purposes. That's why I chose this path as my career. I am a bioengineer by training, but I'm in dermatology as my main appointment. So there are 2 main approaches that we take in the lab to engineer human skin. One is reverse engineering approach, where we start with a skin biopsy. We isolate the cells, each cell type individually, and expand them individually in vitro. Then we use 3D bioprinting to put them in hydrogels and to reconstruct them in 3D. The advantage of this approach is that you can plug and unplug each cell type of interest for the application. But then if you want to really match the cellular diversity of the skin, it gets really complicated technically. So that gap is filled by forward engineering, where you start with pluripotent stem cells this time, and you put them in cellular aggregates. You treat them with extrinsic factors to induce organoid formation. This gives you a lot of cellular complexity, but then you don't have much control over the organization of these structures. So our lab takes both approaches. So why is bioengineered skin important? It is one of the major application areas is drug testing and disease modeling. So here I'm showing you the number of drugs discovered per billion US dollars in R&D spending over the course of years. And this sharp decline basically tells us how much more we pay each year to discover drugs. And the reason for this, now we know that 95% of drugs work in early preclinical animal tests, but then fail in later human trials. So that means we are really much better at curing diseases in animals than we do in humans. So this fact was recently acknowledged by the FDA, who no longer requires testing of drugs on animals prior to clinical trials. So this makes really the 3D skin models, not only skin, but all the organ models highly important. Another application area is skin transplantation. Engineered skin has been grafted for many years onto patients with large wounds. These are mainly burn or traumatic wounds, and more recently, patients with genetic diseases such as epidermolysis bullosa. Skin is usually referred as the first bioengineered organ. Thanks to the studies, pioneering studies back in 1980s, where they first isolated cells, keratinocytes and fibroblasts, and cultured them in vitro, and reconstructed them in 3D hydrogels, typically collagen type 1. But even today, when we talk about commercial or a clinical bioengineered skin, we're talking about 2 or 3 cell types, as opposed to human skin, which has over 50 different cell types, and all these important components, pigmentation, hypodermis, vasculature, hair follicles, sweat glands, and different types of sensory neurons and immune cells. So in the past, our work included improving the cellular component of the human skin by integrating all these important components such as pigmentation, perfusible vasculature, different types of immune cells, each sensory neurons, and hair follicles. But as I was working on these projects, and when I started my lab, we got really interested in one component that we really overlooked in years, which is the geometry component. So here, I want to show you a typical process that is still used today to generate 3D skin in vitro. So we start by encapsulating dermal fibroblasts in a hydrogel that is typically collagen type 1. These cells remodel the collagen gel, and as they do so, they contract the gel significantly in all sites. And then on the top surface, you see your keratinocytes and bring the tissue into air-liquid interface culture. That means you have medium on the dermal side and you have air on the epidermal side. And usually what you end up with is this circular or rectangular tissue that has open boundaries on all sides. But when you think about human skin development, our skin does not develop as these individual patches that integrate with each other later on. It is rather from the very beginning a continuous tissue and is fully enclosed, in which there is a balance between the mechanical balance between the cells and their extracellular matrix. So at this point, we hypothesized that if we can recreate this 3D environment, we'll be able to generate more physiologically relevant skin. And in addition to that hypothesis, that there's important clinical relevance here as well. So today, with these circular or rectangular patches of skin, you can really be successful of grafting them on flat body parts. But when it comes to grafting them on more anatomically complex parts like your hand, it gets really challenging surgically. So we envision that if we can make the skin in the shape of a hand, like a skin glove, the patient would just slip their hand into the skin glove, and then you would just have to suture around the wrist area, and that would be the surgery. So in the past 4 years, we worked on this, and we came up with this method, which includes 3D printed scaffolds. So we made this skin around these different scaffolds. Today, I'm going to show you 3 shapes: the simple cylinder, the mouse hind limb that we use for transplantation, and human hand. So we first characterized the epidermis component by looking at the different differentiation markers. So to show that we have a proper differentiation of the epidermis throughout this shape. And here we applied fluorescently tagged molecules in different regions of the hand, and over time we collected the medium and measured the concentration of this, and plugging into computational models, we were able to estimate the permeability of the epidermis, so it gives us the possibility to study drug transport through epidermis. Then we looked at the dermal component and compared the wearable to the conventional way of making the skin. And what we found was, once you put these cells in this 3D enclosed environment, they start producing more of the dermal relevant ECM compared to the conventional. Next, we wanted to see if this really is reflected in the mechanical properties. Then for this, we took biopsies from the wearable and the conventional, and we collaborated with Dr. Myers and Kaiser lab in mechanical engineering department. They are experts in stretching soft tissues. And once we did applied mechanical tension on this, we found that both the rupture stress and the tangent modulus properties were significantly enhanced in the wearable compared to the conventional. So in this slide, I want to show you a feature that we really got fascinated about when the first time we saw this. So here we're trying to make the skin in the shape of a mouse hind limb for transplantation purposes. But before we transplanted onto mouse, what we did was we took a 3D scan of F-actin molecule throughout to see the cellular and ECM alignment. So basically what we found was, depending on where you are around throughout this complex shape, your cells and ECM are aligned in different directions. And then we confirmed this with second harmonic imaging and elastin staining. And in addition to that, we took biopsies from these different regions. We stretched them in parallel to these fibers or in orthogonal to these fibers to see if we'll capture any differences in mechanical properties. And what we found was we really started to capture these mechanical anisotropic mechanical properties of the human skin that we never seen before in 3D skin constructs. So that was really exciting for us. So finally, we wanted to vascularize it because from our previous experience, we know that when we graft these onto mouse, you need some vasculature, existing vasculature for them to stay viable on mouse. So for that, we introduced endothelial cells that are GFP tagged in this example, where they accumulated in the pores on the scaffold first as these isolated islands, from which then they started making these interconnections. And once we grafted this onto mouse, we showed evidence of anastomosis. And finally, can we run the video? Or should I just, oh no, I didn't do it that way. Okay, so finally we were able to graft this hind limb shaped skin onto mouse, and the grafting procedure was under 10 seconds, and placement was under 10 seconds. The whole procedure was under 10 minutes, and this is a really difficult to graft area when you work with rectangular or circular flat patches. And then we did a lot of characterization to show the proper integration of the tissue. And with this technology now, what we're trying is to go into larger animal models like pigs, and then towards translational studies and clinical trials. Another direction that we want to take is the integration of the wearable skin with the vascularized composite allografts for limb and face transplantation. We are also interested in mechanotransduction in fibrosis and aging, since we think that this is a model that can mimic the biomechanical interactions of the cells better than the previous models. And finally, we want to adapt this platform for drug and cosmetic testing. And this is an unpublished study, very recent version of what I showed you. So basically, we miniaturized it significantly, and we got rid of the pumps and tubes. So this is a standalone device that can be used for drug testing or disease modeling. So last slide, I'd like to acknowledge the people who worked on this and the other lab members, Dr. Alberto Popilardo, David Alvarez. They're both medical doctors who led the project. We are a very diverse group of people. We have from medical doctors to bioengineers to electrical engineers, and I want to thank our collaborators, Dr. Miles Lab, Kaiser Lab. They have been great collaborators, and the funding agencies. And thank you.
So our final speaker, last but certainly not least, is Dr. Ed Guo. Dr. Guo is a Stanley Dicker Professor in the Department of Biomedical Engineering, as well as our previous chair. And so he'll be discussing bone biomechanics and mechanobiology.
Thank you, Alice. It's great to actually present. I haven't presented for a long time. So it's great to be back as a professor. So I'm going to just talk about one story. Hopefully I can get the slides going. It's about the bone and cartilage in osteoarthritis. So I have to emphasize, by my English learning, the things in front is actually more important. It's osteoarthritis. So I don't have to go to much detail, but it tells you it's really tremendous challenge. And then recently, it's not announced yet, the NIH DARPA put a call, essentially want to making a whole knee to solving this problem. You can see one of the trouble we have right now, you don't have an early diagnosis, and then also you do not have any treatment available right now until you go see the orthopods. And then our colleagues doing tissue engineering have not provided the solutions yet. So I have to tell them, maybe we're looking at the wrong place. We should look at the bone, because it's called osteoarthritis. So the thing is changing everywhere, right? When you get to end stage, you have a erosion of cartilage, and then everything happens. You have osteophytes, you have inflammation. So it's a big mass we're having here. But the sequence pathology is still not known. So that's the cartilage and the bone units in the joints, right? You can see this is a micro CT images. You can see there's a cartilage in here. There is a separation between the cartilage and the bone, so that's called subconjugal plate. And underneath them, that's my specialty. I studied that spongy bones you have in here. So the challenge we have right now in the early osteoarthritis, you measure the cartilage, they're normal. You look at the bones, looks normal. So therefore, that's why we have been debating before I was born, and then whether it's a bone disease or it's a cartilage disease, right? So that's why, because when the bone is normal, cartilage is normal, you're fine. But at the end, when cartilage is not normal, bone is also not normal. So that's the challenge we're having here now. So you can see cartilage definitely changes with OA. You can see these healthy ones, and then this one is beginning to degenerate. You're lost in the color because you lost the proteins in here, and they can surface become not smooth, right? The property definitely changes. And then changes in the subconjugal plate as well. So that's a piece of quite a porous plate, and then the thickness increases, and then mineralization decreases, stiffness decreases. The thing changes in here. So come to the bottom of that one is trabecular bone. That's my specialty. Okay, I've been studying osteoporosis for a long, long time, and then 10 years ago I decided to do something, to do in relate to cartilage. My colleague. So we developed this technique. It's called a individual trabecular segmentations. Essentially, the trabecular bone looks very complicated, but it's very simple. It's just bunch of we call them plates in red and the rod in green. So in red is bad. That's why green is good. So that's why in rods here. Normally, in femur, in your spine, your bone is actually organized. You can see the plate are dominant in number, but most plates are in the axial directions, okay, in the loading direction, because they need supporting the load. And then your rod is opposite, is in transverse directions. They are less mineralized, and then also serving as really connecting the plates together. So that's normally what happened. So when the first time I have my exchange student from Hong Kong, he did a study of subconjugal bone. I say, you must have made a mistake. We never see the plates distribute like this. You should be just here and in here. And also, in the subconjugal bone plate, it's actually quite different. It's mostly dominated by the rods in here. So, and also in the plates, you can see in the subconjugal bone, and then they're actually isotropic. It's very different from the femur and anywhere else. And then you can look at it. The modules in here, this subconjugal trabecular bone, this femoral neck, is quite different, right? So therefore, which makes sense. You need to transition from very soft cartilage to bone. You cannot be too stiff. That's why the bone have to be appropriate. So we found out many years ago, identified, because we do these plate rod segmentations, and we found out maybe bone changes earlier. So this is a typical samples. Both are from osteoarthritis, because when you have a total knee replacement, and then you get a sample. When you get a sample, you can see some places the cartilage looks normal, the bone also looks normal. This is at the end stage of OA. You can see the cartilage completely not normal. I mean, the surface becomes, look at the bones I have here. They have a lot of bones, right? But as I mentioned before, I want to re-emphasize, if you measure the property of this cartilage in here, they're normal. Okay? And if you measure the cartilage here, it's not normal. And then if you just measure how many bone volume factors in there, they're actually also normal. But it's because if we're able to separate in plates and rod, we call them controls and intact region, but both OAs, right? Damaged region. You can see in the intact region here, they really have lots of number of rods in here, and plate thickness begin to increase. Because you finally tune into plates and rod, that's where the difference shows up in here, right? So, and then it's also so the plate and rod market structure changing may be proceeding in the damage and also progression. Why that's good news? Because we know a lot of bones. And then there's tons of drugs available to treating bones this way and that way, right? That's what I hear. But interesting, nobody really had been looking at cartilage and bone together. So we're separating bone people and cartilage people. Nobody actually testing cartilage and bone together. So this is a very simple study I want to present it, and then you can show you, we're going to do some cartilage structure and cartilage mechanics, and also bone structure and bone mechanics, and also linking them with OA. So what you do is, so we're going to quantify both structure changes and tissue properties, and in both the OA and the control samples, but also we want to look at how the cartilage and bone are related to each other, right? So this is when you have osteoarthritis, we can go to the surgery department in here, get the tibia plateaus out, and then we're also getting some collections of samples in here. Now you can take in some samples, osteochondral plugs, and you can study them, okay? And then we study them, of course, doing the macro CTs, we can quantify structure, we can also quantify the mineralization. And then you can look at the subcondylar plates, segment them out, and measure its properties. So I'm going to go to details per se. And then this is the key, right? So we're taking this subcondylar trabecular bone you have in here, and then you segment them into, say, plates and the rod, right? So you can quantify how much plates you have here, called plate bone wall fraction, how many number of plates, thickness, and then also very important thing, it's called PR ratio, okay? Plate versus rod ratios in here. Right? And then we can take the cartilage. You can also measure the mechanical property of cartilage as well. You're going to show the results. And then you can do histology. You can see here, there is a control samples have full thickness of cartilage, beautiful. This is very end stage OA. You can use this one, quantify, as well as classify them whether they're normal or not. Right? And then we'll also do micro-indentation test. You can indent them on the subcondylar plate in a custom-made micro-indenter in the laboratory. And they also, you can go inside the trabecular bone, making indents in here on the plates and rods, as well as different orientation. It's pretty tedious experiment, but actually you can quantify everything you want to do. So now, look at some results. So on the left side, those are control samples in the media and lateral. So the top are medium side, the bottom are the medium side of the knee, right? On top one here, you can see on the lateral side, we have 36 samples, and the medium side also 36 samples. But of those 6 samples, if you classify by the histology, you find out about 28 of them looks normal, okay? We call them moderate OA. It's not exactly normal, but a moderate OA. And this one, 34 of them in the most median side is called them severe OA. So indeed, if you look at the properties right now, if in those moderate OA, if you do the classification by histology, that's how you quantify whether I have a osteoarthritis, they're actually completely normal. But in the severe one, certainly the score is different, right? And interestingly, if you measure the thickness of cartilage actually is quite sensitive. In the moderate OA, you already have significant reduction in the cartilage thickness. So the thickness is changed. But if you look at the density of the tissue, you may have some changes in the chondrocyte density in the early moderate OA. But the GAG content actually is no different. So that means the biochemical contents of the cartilage actually is not changed. And as I mentioned before, in the moderate OA, there's no cartilage tissue property change until you have severe OA. You can see here, the severe OA, you have reduction in mechanical properties in here, right? So therefore, but if you look at subcondylar plate, and then there are actually significant changes in both moderate and severe OA, the significant increase in here in terms of thickness. And there also significant increase of the mineral density, tissue mineral density. And also, interesting, the early time, you actually have significant decrease in tissue property. So something happens in the bone, actually much more sensitive than in the cartilage in here. And then if you look at trabecular bone, as mentioned before, in the individual trabecular plates and rod, you both have significant reduction in the modules, even in the moderate OA, that persist in the severe OA as well. And then those not totally explained by the tissue mineral density yet. And then because in rod, the tissue density actually is increasing for the rod. Okay, so that's probably because the surface, we indent on the surface of trabeculi, and then those probably in the surface different. Yeah, it's going to be ending very soon. So And then if you look at the correlations, everything together, that's actually something interesting. So this is a control sample, normal sample. You can see if you plot how related to osteo score, and you found out bone one fraction, plate bone one, rod bone is negative correlated, because it means you have soft density, then you have good bones. And then if you have heavy bones, you have a heavy cartilage, right? And then this one, interestingly, begin to lose when you have moderate OA. And then all the rod ratios, rod property actually is not related to the scores. And then if you're in severe OA, they become totally independent. So essentially, it's two separate tissues in here. So I'm going to end up in here. And then, so this is our theory. Called OA initially in bone. There's something if you can quantify thing here, and then if you're preventing this one happens, definitely is going to preventing getting the later stage of the advanced OA. So we just kick off the good thing is you actually can study in humans, not very convenient. So this is me scanning these high resolution CTs, and this is my knee. Looks beautiful. And then I just repeated my study in December, and then my student just have a follow-up analysis of me over 7 years, and my knee looks great. My plate rod ratio actually decreases, so I can confidently say with n equal to 1, I'm not going to have articulate cartilage. So at the end, I'm going to thank you. Oh, I'm missing one slide. Okay, yeah, it didn't change. So I want to make a shout for this training grant, and for those people who are U.S. citizen and permanent card holders. And if you're interested in this training grant, please talk to me. Talk to us. Yeah.
So if all our speakers can come up, we can have about 10 minutes for questions.
So, Dr. Guo, so do you think that since it appears that bone is impacted first, that it's primarily a mechanical signaling that is what damages the cartilage, or do you believe that there's like some kind of inflammatory secretion or some kind of signaling coming from the bone to the cartilage? How would you say that the primary mechanism from one to the other? Yeah.
Yeah, so I think, actually I can explain it. The two risks you have osteoarthritis. One is overweight. So I can also explain why overweight causing the OA, not only just the cartilage, because overweight also causing the loss of bones, because you actually have micro damage and then you loss in bones. And then another big factor, which actually we're going to also study, is ACL tear. So ACL tear essentially is a death sentence for your knee. Okay, so 10 to 15 years, you're going to have a high chance of osteoarthritis. And then what happens is in that acute phase of ACL tear, you have loss of bones, tremendous loss of bones. And because you have inflammation, and also you have unloading because you on the crouches. So when you do that, we have quantified already, you're losing the rod. Essentially, even though you lost in bone, you can recover a little bit, but the rods are gone. So we think, this is, if we're able to explaining that, and then the first thing we want to do is able to providing this as early detection and also has mechanisms for treatment, because we have a lot of bone drugs available to preventing this bone loss.
For Dr. Chahine, so looking at this delivery of MSCs in this pro-inflammatory environment, so you're seeing some recovery of these pro-inflammatory or anti-inflammatory markers like IL-10. Are you seeing actual implantation of these cells and regeneration of the tissue, or just kind of a reversal of the the pro-inflammatory environment?
Yeah, that's a really important question. So it's been debated as to whether the cells are working in a way to be a replacement cell source to then promote extracellular matrix production or act in this anti-inflammatory way. Our data suggests that they're really just quiescing the inflammatory response more so than playing a role in being the replacement cell. However, there could be some crosstalk or cell-to-cell contact communication between the MSCs and the disc cells, which then promote the disc cells. So, but we don't believe that their job is to make this replacement ECM.
I had a question for Dr. Nurker. So I was really interested that within a species, the number of loops is so stereotypical, but that they're different across species. And I was just wondering, like, how does the number of loops regulate its functional properties of how the animal digests? Is this tied to diet? Is there anything known about this?
Yeah, so I think the selection pressure is the diet that's available. And so it's more, how long of an intestine do you need based on the resources you have available, and then can you package that within the viscera? And so, like, marine mammals, for some reason, have just insanely looped intestines. Like, if you look at a dolphin or like a elephant seal, it's like the loopiest of intestines. But in general, like carnivores that have very calorie-rich diets have shorter intestines. Herbivores have larger intestines that have to work harder, and so therefore are more convoluted in their loops. So I think it's a combination of selection pressures on the diet and the geometric confines of the body cavity.
Thanks. Sorry, I'll stand up since I couldn't see you very well. But this is a question for Dr. Guo. I was really interested in your individual trabecular segmentation methodology, and I was specifically curious about what the advantages of that methodology over something like a micro-CT, and also how does the methodology distinguish between rods and plates? I just want to know more about that.
Yeah, so the technology is based on micro-CT or HRPQC for the human scans. It's based on the one. But the technology is based on topology analysis of this structure. So essentially you're separating a two-dimensional structure, which is surface, and then one-dimensional structure, which is the rod. And then it's, I think I give my research career. It starts with fun. Because I thought I know mechanics, right? Plates and rods are different mechanics. So I don't know imaging when I was doing my postdoc, and then I decided to pursue this to say, computationally is much easier. But turns out that's actually quite important because now we don't do too much tissue mechanics anymore because tuberculum mechanics is done. So essentially that part of my career, I essentially put a nail in my own coffin. So, and then now, of course, in terms of osteoarthritis, it's really tremendous interest.
Great, thank you.
We have one question here.
Hi, this question is for Professor Guo. I was curious if you've done any research into introducing or completely removing hormones within your studies, such to replicate situations such as postmenopausal women.
Yeah, so we have done some simulations, really look at the contributions, of the simulations. We can remove plates and rod. You will find out in the osteoporosis, if we remove rod, doesn't do too much, and then because most contribution come from plates. And another thing is also interesting is there are people has differences. So Chinese woman is dominated by plates. And the Caucasian woman is dominated by rods. So there's a huge tissue between them. So I actually have studies in China now. I launched them in December 2019. So my timing is perfect. So I haven't done too much. So in a couple of weeks, actually, I'm going back in there to rescue that project. Because I have a RV with this HRPQC in there. So my dream is driving this truck to all different exotic places, and they have studies done.
Thank you.
Hi, my question's for Dr. Abachi. Very interesting. I was going to ask, have you tested how the wearable skin is going to change over time? Or do you have any predictions on how the transplanted skin would change in comparison to the original skin?
Yeah, great question. So what we've done so far is done on mice, and the longest we tracked the cells and the tissue was 4 weeks. We kept it at 4 weeks because that's when you start having revascularization, a little bit of innervation also in the tissue, and we stain for human-specific markers to make sure that the graft, because the graft is made of human cells, to make sure that the graft is indeed still there with all the human cells that we initially grafted. But we are now planning on more in-depth studies when we do it in pigs. In that case, we'll definitely label the cells in advance, and we're interested in seeing where these cells end up and how long they'll last in the tissue, and if they're going to be, for example, taken over by the surrounding cells, or they'll stay longer, is something that we want to trace in pigs, because it's more important in pigs because pigs can heal similar to human skin, and mouse skin is not much. It's more contraction rather than reepithelialization in mice.
Thank you. I also had a question for Dr. Abachi. So we talked a little bit more about like the hind limb scaffold that you had shown, but for the glove application, what are some methods, or how do you envision, like the skin on the palm is very different than the skin on the top of the hand. Like how would you be able to control that when growing the scaffold at the same time?
Right. It's a very, I would call it a difficult challenge to create the palm skin. Nobody has been able to achieve that yet. It's still a question whether it is intrinsically programmed, genetically programmed during development, or is it microenvironmentally controlled. When we're first born, our palms are not significantly thicker than the other sides of the skin, but over time, as as the babies grab on things because of the friction and mechanical forces that are coming extrinsically, they develop thicker epidermis and start expressing different markers. But some of those markers are also coming intrinsically, even at birth. So far, what's been done was there have been groups which isolated palm keratinocytes, and they tried to develop in vitro models using those cells, and they were able to show those markers. I think it's a combination of environmental and genetic factors. So if ever we would create a real palm, I think we'll need cells coming from those anatomical sites. That's what I think, but we've never done that yet.
Thank you.
I have a question that basically just continues on top of that. It might be a very similar answer, but her question was like palm versus back of hand. What about like different populations of people? Just like skin in general, like my skin may be different than yours, which may be different than somebody else's. Like, is that going to be the same process as like the back of the hand versus the palm, or is that like something else you would have to look into?
I don't know the answer to that question specific to the palm versus the back of the hand, but I know that our skin is definitely different between people, but not only between people, even the different anatomical sides. Your facial skin versus your skin on your forehead, forearms. They're all the expression of the cells, the gene expression profile of the cells in the dermis, especially dermal fibroblasts, they are different. So I don't know about the clinical relevance of that question, but if we want to transplant these engineered skin onto patients, it should come from, at least now, it should come from autologous sources. To avoid any immune rejection. So your concern may be addressed by that.
Okay, thank you.
Have you considered which would be the injection factors of the graft?
Injection? Sorry, I couldn't. Was it for me?
The rejection.
Rejection. Mm-hmm.
Of the graft. Did you consider which circumstances will be causing that, and how to avoid or how to prevent?
I mean, there are products out there, and there have been studies also using allogenic sources of cells to make skin, and in those cases, most of the time you have immune rejection in the patient, and those are not ideal grafts for patients. That's why we want to go in the path of autologous cells. But the disadvantage of autologous is obviously when there's a burn patient, for example, if it is an acute wound, then it takes us 2 to 3 weeks to develop these engineered grafts. So in the meantime, this patient have to be covered with something temporary. And then after that, whenever our engineered skin is ready with the patient cells, we can graft it. That's not the case with allogenic. That's why you see in the commercial area there are more allogenic products than autologous ones, because they can make it off the shelf ready for the patient. But eventually, I'm hoping that will reduce the time to make the skin from patient cells. Right.
I have a question for Dr. Guo. So one of the reasons that you mentioned for investigating the role of bone in particular for osteoarthritis is the amount of treatments that are available. But you also mentioned that an important component of that of the bone morphology with osteoarthritis is like the plates versus rods. Are there any treatments that are specific to those particular components of bone, or is that something that would have to be developed after this investigation?
Yeah, so the plates and rod, it's when you're born, most of your bone is actually rod, and the plates are developed for the mechanical loading. So therefore, the difference between the Chinese woman and the Caucasian woman is also developed during the puberty time because it's mechanical loading. So in the subcondylar trabecular bone, you have a lot of rod just by, because of the loading condition. I think it's related loading condition when you're growing up, so they need to have this transition in there. And then in the pathologic cases, either obesity, have ACL, then you disturb that one, so then you just embedding the things in there. So therefore, for example, I think I always, if I do this, I'm going to retire. Is when you go to a dentist, you do the root canal, right? They give you antibiotics. Right, so because that's actually preventing you have endocarditis. Because before this one, they know if you have a dental procedure, you have a risk of heart disease, you die. Right, so now you can. So I think ACL. I think there is a hope in the ACL in the future. As soon as you have an ACL tear, we give you a shot of anti-osteoporosis drug. Then you will never develop this risk for ACL. I think that would be fantastic. And then doesn't make any money. But because it's using existing drugs available for new use, but the impact is tremendous.
So we have a virtual question, actually two. If a person over 70 years old has an ACL tear and meniscus tears, how fast will the arthritis develop? And is your arthritis treatment available to the public?
Oh no! That's a study we have. We just have patients number 1 this week recorded. So for if you're not treated for ACL after acute phase, then you're pretty much you're done. I don't think there's any technology available right now. So that's unfortunate. So I know I interview many students tomorrow. Very interesting. From those I think 10 students, 11 students, 2 of them have ACL tear. May not save you. But we can working together to find a solution for other people. Yeah.
So with that optimistic, I just want to thank all of our speakers again for really beautiful and impactful presentations. And this closes